we are going to be, I'm not going to be going through the details of them, but I do expect you to understand, the implications for chromosome number after meiosis versus mitosis, because we're going to talk about it when we look at life cycles, particularly of the yeast, Saccharomyces cerevisiae, which we're going to look at in some detail. So you do kind of have to have a kind of a basic understanding. Well, since we don't have a nuclear membrane separating the DNA, and we can't have a nuclear division that would be like what we'd see in mitosis or meiosis, then what's cell division like? Well, we basically do have some similarity in what has to happen in order for cell division to occur in prokaryotes. If we're going to try to compare them to eukaryotes. So one thing that has to happen is in prokaryotes, you definitely have to have replication of the DNA before division of the cell can occur. And that would be so that you could maintain in the daughter cells the same DNA makeup that you would have in the original cell. So remember in the cell cycle, there is an M phase in the eukaryotic cell cycle, which that's the mitosis part, but that's really only one of the stages, and the M phase, the mitosis phase. Another phase called the S phase, and that's the phase during which DNA replication occurs, right? So that has to actually precede the replication of the DNA in the eukaryotic cell cycle has to precede the actual mitosis. And if you remember, there's also the G1 and G2 phases, you can use to round out the cell cycle in a eukaryotic cell. So in common with eukaryotes, prokaryotes have to replicate their DNA, obviously. So DNA is going to be replicated, and in general, for most prokaryotes, a parent cell is going to give rise to two equally sized daughter cells, and we call that process binary division. It's not the only way that prokaryotic cells, divide. There are some other processes, but that's the most common one. It's generally called binary division because it's structured, a parent cell, the original cell that has replicated its DNA, and it divides into two equally sized daughter cells. Well, obviously, it's going to be a little bit more complicated than that, and as people study cell division in prokaryotes more, they come to learn about the role of certain kinds of proteins in the process. We'll talk a bit more about that later on. All right, what about ribosomes? All cells have ribosomes. They need them because protein synthesis is a common function for cells. But if we look at the composition of the ribosomes, in general, they consist of protein and RNA. And those RNAs that are found in ribosomes we call ribosomal RNAs or rRNA, as opposed to mRNA and tRNA, which have other functions in cells. So we've talked about ribosomal RNAs already, and we said that the ribosomal RNA that's found in the small ribosome subunit is one that has been used a great deal to try to get ideas or develop ideas about the evolutionary relationships among different organisms. But if we just look at ribosomes in general, we recognize each ribosome has two subunits, large and small. And for prokaryotes, the whole ribosome is generally described as being 70S. S is a constant, it's called the Steinberg, which is a reflection of how the ribosome behaves when it is centrifuged at high speed. The eukaryotes have what we call an 80S ribosome. So this is the whole ribosome. So prokaryotes 70S, eukaryotes 80S. So what's that telling us? It's telling us that the behavior of prokaryotic ribosomes is a little bit different when we centrifuge them at high speed when compared to eukaryotic ribosomes. Okay, so if we look at if we were to take ribosomes purified and separate the large and small subunits, what we find is that prokaryotic ribosomes have a large subunit that's 50S and a small one that's 30S, whereas in eukaryotes you have these values that are a little bit different. Notice that even though the large is 60 and the small is 40, the whole particle doesn't behave like 100S. So that's kind of just something you have to be aware of. Just like 50 and 30, well, that would give you 80, but that's not how it behaves. So in terms of that particular concept. You can see the table for some details on this. Here you actually see they're actually giving you information about the lengths of the ribosomal RNAs in the different subunits. So bacteria and archaea that have 16S ribosomal RNA in their small subunit. You can see how long it is by looking at this chart, how many nucleotides long. You can see that in eukarya, that small subunit ribosomal RNA is 18S. Again, that same designation based on that constant that I told you about. And notice that the nucleotide length is longer. Now, here's something really, really interesting. If you look at the three domains, even though both bacteria and archaea have 70S ribosomes, and in their small subunit they have 16S ribosomal RNA, if you actually look at the sequences of the ribosomal RNAs and some of the ribosomal proteins in the archaea, what you find is that they are more similar to those of eukarya than they are to bacteria, which is why under protein homologies they say eukaryotic for archaea, and for ribosomal RNA homologies they say eukaryotic. So you're seeing an example of how the archaea are somewhat aligned, have some similarities, important similarities, and we will see more of this. And this is actually part of the basis for why, when the phylogenetic tree is drawn, you see a divergence between the bacterial line on one hand and then another line, which then later diverges to give archaea and eukarya. You may not remember that, but the first, I think maybe the second day of class, I showed you that very simple diagram from chapter 1. But that was the only thing you could see in that diagram. Division into two subdivisions, and then the second subdivision, one was bacterial, and the second one was subdivided into eukarya and archaea. All right, now I have another little bit of information down here about the ribosomes that you find in mitochondria and chloroplasts. And as you probably remember, the ribosomes, or the chloroplasts and the mitochondria, are thought to have been derived from prokaryotes, specifically bacteria, that took up residence in other cells. And this is referred to as the endosymbiotic hypothesis. And part of the support for that hypothesis comes from the fact that these organelles have ribosomes, and their ribosomes are similar to those of prokaryotes, specifically bacteria. So that's just a little tidbit of information that you might know. All right, this is, as I pointed out here, this table, I shortened it here, and they, for some reason, decided to leave it out of the 14th edition, but I kind of liked it, so I put it back in. This is the longer version of the table, and I'll have more to say about this table later. I just wanted to show you, okay, I made a shortened version. There's the original. All right, what else can we say about ribosomes? Well, we went through the fact that bacterial ribosomes are different from eukaryotic ribosomes. So if we have a bacterial infection, if we could find a drug that would specifically inhibit the bacterial ribosomes and not the ribosomes that make up, most of our cells' ribosomes, and yes, the mitochondrial ribosomes would be susceptible to some drugs that work against bacterial ribosomes, but most of our ribosomes are in the cytoplasm rather than being in the mitochondria. So the protein synthesis interference with the function of the ribosome is a good mechanism or a good way for trying to find drugs, antimicrobial agents that specifically work against bacteria. And there's actually quite a large number of antibiotics that work like this. So tetracycline, chloramphenicol, streptomycin, there's a whole slew of them, a whole long list of that. All right, so this is a kind of simple model of a eukaryotic cell, and you'll notice that it has quite a few additional structures, right? And guess what? I'm going to let you review those on your own, except for a couple. And the ones I want to spend some time on, I'm going to spend a little bit of time on mitochondria. And this cell is, yes, it is photosynthetic. It's got a chloroplast. So we're going to spend a little bit of time on mitochondria and chloroplasts. And we're also going to spend time on some organelles that appear to be degenerate mitochondria. Why do you suppose we're going to spend time on those? Because they came from bacteria. And our bias in here is really toward the prokaryotes. And yes, we're going to talk about the eukaryotic microbes, but these other organelles, you can review those on your own. But I do want to talk about mitochondria and chloroplasts. So let's visit the mitochondria. You probably remember that in cells that are carrying out aerobic respiration, suppose they are given glucose as a substrate and they break that down into glycolysis. So after glycolysis they have pyruvate, and then they take the pyruvate and react it with coenzyme A and get acetylcoenzyme A. Then the acetylcoenzyme A goes into pyruvate goes into the mitochondria, where it reacts with the coenzyme A to get acetylcoenzyme A. Then the acetylcoenzyme A reacts with and enters into the citric acid cycle. So in the citric acid cycle, you basically have a cycle of biochemical reactions, and at different reactions on that cycle, you generate reduced electron carriers, NADH, FADH2. Those are going to donate their electrons to an electron transport chain. Electrons go down the electron transport chain, and as that happens, hydrogen ions get shuttled across the inner mitochondrial membrane, this membrane here, establishing the proton molecules. If you don't know this now, or if you're not relating to this now, you will by the time we get finished with metabolism. But, if you're at the point in your studies that you have gone over aerobic respiration as it occurs in chemoorganotrophs numerous times, you may just kind of be with me at every step of what I'm saying. I don't know where you are in terms of your studies. Some people think you've gone over this multiple times. Someone in here could have only gone over this maybe 2 times, or as good as 2 times. I can't imagine there's anyone in here